Hi, everyone. Thank you for putting up with me today. So today I'm going to talk about our work on parallelizing a image analysis over a large set of very large images. So we've been working with whole slide images, which means the microscope scans over an entire slide, which results in these tissue sections that are about 40,000 pixels on a side. You have 2 to 5 of those per slide, and we've got hundreds of these slides within a data set. So we're dealing on the order of 1 trillion pixels per data set, which, as you can imagine, has some compute limitations and requirements to deal with. So our kind of steps that we work with is we first need to grab the images off of our image server where they're stored. We then convert them to ZARs because microscopy format images are not great to compute on. Then for this project, we segment out ROIs. In this case, it's the glomerulus, which, if any of you know about kidney biology, it's the filtration unit. So it's what takes the stuff you don't want in your blood out. And we then upload those ROIs for storage and pass them along to our feature extraction pipeline, which uses a truncated pre-trained network. And we operate across all of our ROI classes, and then we also take random tissue patches from outside of the ROIs so that we can kind of cover our bases and find anything that might be something we couldn't segment but is still biologically interesting. So we kind of tried a number of things to get this working at scale. What we kind of decided by the end that we really needed was multi-language support. The pipeline actually has a few different language steps in it. Needed to be easy to implement in terms of we'd written the actual analysis as bits and pieces that we wanted to be able to wrap in something that would then properly handle the parallelization for us, and we didn't want to have to do too much rewriting of our original analysis code. It needed to have good data handling. There are a number of steps in this that we want to be able to effectively pipe the data in between those steps. And it needed to be able to work with very large files. So our first attempt was with multi-processing pools in MATLAB, which is what we were originally using. That didn't really work. They are, frankly, very complicated if you're trying to implement them for something that doesn't need them. And you end up losing a lot of work if it breaks partway through. Unlike, say, NextFlow, you can't just resume the process. There's some checkpointing, but it's not nearly as good as what NextFlow provides us with. Our next port of call was DataJoint, which is a software solution that relies on a relational database backend. It only works with MATLAB and Python, or at least it did back when we were using it. It might have expanded since then, I'm not sure. It doesn't handle large files well, and depending on exactly what you're trying to do, massaging your code to work with that relational database can be complicated. Finally, we were trying to go back to basics. We use a batch APC with Slurm submission, so we decided to let's just try job arrays. That is a very simple way of doing naive parallelization. And it solved a lot of our issues, except that it requires a great deal of bookkeeping on our part. I think after the training yesterday, I feel it's a little unfair to say that they're effectively the same as NextFlow without handling the bookkeeping, but that is what we're using NextFlow for. So we're hoping to implement some of the more advanced tools that NextFlow provides going forwards. So again, NextFlow really was suggested by a colleague, and it really solved all the issues for us. So this is a kind of 1,000-foot overview of our pipeline. We grab the images and convert them separately because we actually use those for a number of downstream analyses. Then we do the segmentation, as mentioned before, identify the ROIs, and go over each of the ROIs and extract features. Of note, there are a few different levels at which parallelization occurs in this pipeline. First off, some processes just need to be done sequentially. This is mostly things that interact with the imaging server because I actually broke it once trying to send too many requests at once. So since then, we limit that to being sequential processing. We can operate on some things at the image level. So we do our segmentation at that level. We farm out each image to a separate process. And then once we actually have the ROIs, we can speed things up by just taking like 20, 30 ROIs at a time and feeding each of those to an individual process. The ZARs allow for parallel reading of the files, so we don't have to worry about any of that. We can't request enough processes to actually increase the number of processes that we're operating on by doing this, but since it makes each individual process smaller and shorter, it makes it a lot easier to kind of fit into the gaps within our HPC's scheduling, and also means that we aren't having things sit around while we wait for the last one to finish, for instance. And then, again, feature aggregation, we need one file out of that, so it's done sequentially. There might be a nice way to do that, but I'm not sure of any way of doing parallel writing to CSVs. In terms of some stumbling blocks for us that we would, if anyone in the room has any ideas on how we could address these, one is that there are times when we have to go back and modify the underlying research code in a way that isn't caught by NextFlow when it is setting up the hash for the resume caching. And it would be nice to have a flag that would basically tell NextFlow, don't resume this process, always restart it. What we've been doing so far is we create these channels that are basically just iterated to, their whole purpose is to modify the hash so that it knows to redo that process. But if anyone has a more elegant solution to that, we'd be very interested. Finally, it would be nice to have a bit more, be able to provide a bit more information on the hierarchy. So like in our case, each of the ROIs belongs to a specific image, and it significantly complicates things when, for instance, we're grouping ROIs and it puts the tail end of one image in with the start of the next image. So some way of just telling NextFlow, don't group across this higher level image or a higher level channel would be useful. Again, happy if anyone has any ideas on that. I would like to thank Matt Mahoney, my primary investigator, and the Corstonia lab who did all of the wet lab work for us, as well as the Nathan Schock Center, which provided the funding for this. Thank you.
Thanks a lot, Seamus. We've got a couple of questions here. The first one is from Ken in terms of, can you speak a little bit more about the details of the segmentation and the feature extraction steps of the pipeline?
So segmentation is done with a UNet. We got a pre-trained one. I forget what it was originally designed to segment, just as a hot start, and then we had a poor postdoc go and circle a bunch of these glomeruli to train it to specifically segment what we were looking for. And it's not perfect, but it is relatively effective, and at the scale we're working at, a few, like 0.01% mistakes kind of get lost in the much larger good data. For the feature extraction, again, we use a pre-trained network that was trained on ImageNet, and we just truncate that. So the kind of underlying idea is that it has been trained to, at that higher level, understand more basic concepts of the image. So like, is it round in this place? Are there straight lines coming across it? That sort of thing, basically the convolutional layers. And by taking them there, we basically have a single vector description of the image that can then be more effectively used for downstream analysis than, say, a pixel values.
And we have a question from Anand. Do you have any of the pipelines available? Is there any repos available?
Currently, no. This project is waiting. So this project was based on mouse aging, which means that once we found something interesting, we then had to wait 18 months to get new old mice to confirm our findings. So everything is currently kind of on hold until we can get that confirmation and can publish things. At that point, we are hoping to get things up on GitHub. Well, they are up on GitHub, but make it a public repo. It will probably take some cleaning up, because up until this point, we hadn't really been using NF Core best practices. That's part of the reason why I'm here, was to learn that sort of stuff.
Awesome. Well, there's something else to learn. Your requests for invalidating the cache. There is ability to do this with a process.cache option, which you can specify in the process. So there's inside of the Slack, there's some good discussion going on there. And a final thing, that just a comment that was coming from Harshal there, is that in terms of the job arrays, NextFlow in the latest release, really, I think it's from a couple of weeks ago, now supports job arrays, which may be really relevant for these very small jobs that maybe don't last very long, that you want to put onto a cluster, sort of combine all that stuff together. Paolo's going to be speaking about that later on today. Awesome. Thanks a lot, Seamus.
Thank you.